Thank you very much, Jose, and thank you all for this wonderful invitation to be here. This has been a really exciting forum. It's fun for me to hear a lot about the freshwater mollusks and land snails yesterday, and reunite with some of my colleagues that work on ocean acidification and bivalves. I guess you'll hear about that later. So I want to talk a bit today about mussels. We know that there are mussels in our body, but the ones I really care about are the bivalves. And I want to tell you a bit about how they attach and why we should care about that, and what I think will happen to them in a warmer, higher CO2 world. Okay, so I am from the University of Washington, and here we are on Sanibel Island, and it's always interesting to me how I gravitate to different islands, and probably this audience does as well. Just wanted to give you an idea of where I'm coming from. So there's Seattle and Vancouver here, and I'm on these in the San Juan Islands in a town called Friday Harbor. So many of you have been there. One of the key points to keep in mind is it's a lot colder there than here. So things are a little bit different. We have mountains, so there's a little bit of a different topography to the waters there. We do have charismatic megafauna. We can count them. So our resident population, we're the only county in the United States that has a population of whales within it, and we have 84 of them this year. But what I want to tell you about is not the cute big dolphins, but what's happening here on these rocky shores. And when I talk about what I have done, what I really mean is what we have done, and I have a number of wonderful graduate students that have worked with me over the years, and here's just some of them, and I will be presenting their work. Only a small bit of it is truly done by myself. And one other point I wanted to make is here we are kayaking in the Pacific Northwest, and I hope you can see that we are clad in all kinds of coats and hats, and it's really beautiful, but it's really cold. So just recalibrate your brain to what it's like to be in colder waters. So what I'm going to tell you about are mussels, because in temperate zones worldwide, they are the dominant species in that mid-intertidal zone. Okay, so we're no longer talking about the deep ocean or the mid-ocean, but that little bathtub ring around our oceans, which is the rocky shores. And they have been studied for many years because they are really accessible, so people notice them a lot. And ecologists have used them as a really important model ecosystem to develop ecological theories. And mussels have really played an important part of that. So they're really the lab rats for ecology. So what I want to tell you about is what I call ecomechanics. So it's a type of physiology, and what I'm interested in is looking at the mechanical limits for organisms. So Brad talked about some physiological limitations. If organisms don't have enough oxygen, then they can't live. But it's also true if organisms aren't physically strong enough to withstand the forces that they see, they get crushed or they get dislodged, and they can't live either. So there are constraints, all kinds of constraints that the environment places on organisms. And I want to focus on those mechanical constraints. So this is a field that's called biomechanics, and usually it plays out right here, where you have scientists that will look at the morphology of an organism and make some inferences or measurements about how it functions. And so this is also known as functional morphology. Biomechanics, there's a whole bunch of, a whole journal of medical biomechanics, and people are interested in designing artificial hearts, artificial knees. I don't do that type of. Oh, I did what you did, Brad. Okay, let's see. What I'm interested in is how things on the rocky shores in nature are designed. Rocky shores are one of the most mechanically stressful environments on Earth. At low tide, they're subjected to a lot of heat and desiccation. At high tide on these exposed shores, big waves coming in and crashing on them. And the organisms need to be able to withstand the hydrodynamic forces that they experience. So I'm interested in how things are designed to put up with those forces, and I'm asking how do they perform in their natural environment. So this relates to ecology, and ultimately I'm interested in how those different structures evolved. And that's where you get into fitness and evolution. Okay, so why mussels? I did say that they're the lab rats for ecologists, but you know, most people don't really care about ecologists or what we're studying or what kind of theories we're developing. So what I wanted to do is to take some time and tell you why we should care. And I think a lot of this boils down to this type of slide where people think about what services an ecosystem can provide. And so I can play this game. The general categories, this came from an environmental science textbook. But the human society values ecosystems, and we can put dollar amounts on these values. If they support something we care about, if they provide something we care about, if they have cultural value, or if they help regulate something about that environment. So mussels do all of these things, and I just like to march through how they do that. First of all, mussels support and structure coastal communities. They attach to hard substrates, they're very numerous, and they stabilize and modify their local environment. So many organisms find habitat in and amongst mussel beds, so they increase the diversity of the rocky shore. And also, they are subject, I guess this comes back to the famous ecological theory. And so I have a slide in here showing a disturbance to a mussel bed, this patch that formed. And if this happens often, other organisms can come in and colonize, and this process of disturbance also increases the diversity of the community. And so this type of process, a disturbance to a dominant organism, is a similar thing to what happens with fire running through a forest as well. So this is a really important concept to keep ecosystems healthy. A little bit of disturbance is actually a good thing. And a lot of that theory has been developed with the mussel community. Okay, mussels are filter feeders. An individual mussel can filter a liter of water an hour. And so this is just a nice visual of that. There's an aquarium here with nice some sediment and food in that water column. It's not very clear. If you plunk a couple of animals in there within an hour, you can see that they can clear that water. And so what they're doing is, one, cleaning the water. So often we care about that. But also they're taking food from the water and putting it into biomass, which then can be food for other organisms that we do care about, things like lobsters and crabs. So lobsters can't pick out individual phytoplankton from the water, but they can eat the organisms that are able to do that. So mussels are also food for humans. So this is a picture showing a mussel farm in the Pacific Northwest. And so the way they grow them, these are just floating rafts, and what they have are these ropes that dangle down into the water column. And on those ropes, the animals will attach. So they would be maybe this big around when they have a good accumulation of mussels. So after about 6 months or a year, sometimes 2, the growers will come and bring these ropes onto their harvesting barge, strip them off the line, send them through these sorts of machines. They will clean them, take off the byssus, the beard that they attach with, and then pack them up for shipment. So typically, with our ability to do on-demand ordering and to ship things anywhere in the world overnight, typically the mussels are going from sea to table in 24 hours. Okay. So I like to say that this is a farm-to-table industry. Okay, mussels also have high cultural value. This is an image taken from Vigo in Galicia on the Atlantic coast of Spain. And so this is the major ria in this community right here. Most of the population lives in and around here. And what I wanted you to know is that this is one of the postcard images of this town. And all of these little dots in the water are mussel rafts. So this is a fishing community. The economy is based on a number of fisheries, and mussels are one of them. They supply most of the mussels for Europe. And it's a point of pride. And so the community doesn't think that aquaculture is ugly. They really value that industry. It's a real part of their culture. So I had the opportunity to visit this area, and this grower here took us out to see his rafts. It was a Sunday. He brought his kids. And I really like to keep this in mind, is that this grower would like to, he has rights to his 3 rafts, and he would like to hand those down to his children. And so part of what motivates me is to keep this industry sustainable for many generations. All right, mussels also have high cultural value. Historically, on the West Coast, here's a micrograph of a photograph of a Coast Salish woman harvesting mussels for a potlatch, a ceremonial dinner. And then here's a couple of slides of one of my personal favorite uses for mussels and the fibers that they produce. But this woman here, her name is Chiara Vigo, and I had the opportunity to visit her in Sardinia a couple of years ago. She's 35th generation artisan who was taught this craft of harvesting the byssus from pen shells, Pinna nobilis. There's really big pen shells there. And so she weaves them into fibers. And from that, she made this particular piece here. But her grandmother, for example, has some of her work has been in the Louvre. And it's she will not do it for money. It's just a labor of love and a craft that she practices. So these are some gloves made of mussel byssus. Historically, these were also collected and used as barter. Barter, as money, to exchange for goods, as if it was money. Okay, so that's why mussels are really cool. Mussels are also a problem. They have given people a bounty of troubles, I like to say. And this is one of the reasons why there is a strong motivation to study them. So mussels, like many marine organisms, love to attach to solid substrate. And I've been to some of these marine biofouling conferences, and yes, there's paint that we can put on that will deter organisms, but there is no way to prevent marine organisms from colonizing a structure if you stick it in the water. No matter what you do, they have not invented anything that's going to make a structure immune to biofouling. So mussels are a part of that. So here's a picture showing as larvae, they can get into little spaces and settle, and then they grow, and then they clog pumps. They grow on outer surfaces. They increase the weight of that structure. They increase the drag on that. It's really a problem for things like ships. Here's some just examples of where it can cause problems. It's a real problem in the cruise ship industry. I had someone tell me for cruise ships, they really have 2 expenses. One is the expense of running a hotel. So you need people to keep the rooms running and to feed them. And the other major expense is fuel. So if you get a lot of animals or organisms growing on the hull, that can increase fuel costs up to 25%. So it's really big business to keep the ships clean. It's very expensive to bring them into dry dock. Every day that they're not on the water, getting money is really critical. Okay, so there's lots of interest in making things not stick to structures. Okay, so that's why I've been interested in mussels. I've been doing this for 25 years. And I've always been picking on the local mussels that are in the Pacific Northwest and in North America. And those are the middleid mussels. The main species that you might be familiar with, these are the tasty ones up here in blue. Middleis edulis is the species in New England. There's a little bit of Troscelis to the north. Troscelis is typically the one we have in the Pacific Northwest. And Galla provincialis is the Mediterranean mussel. Another one I won't be talking about, but I have worked on, is Middleis californianus, and that's really a different beast. I like to think of it as the gorilla of the intertidal. It's bigger, stronger, longer lived. Everything about it is just stronger. Okay, but I'm going to focus on the tasty ones today. Okay, so I mentioned I'm interested in mussel attachment, and that is because a lot of the issues we've been talking about, disturbances to mussel beds, that happens when the attachment fails. The the whole aquaculture industry relies on strong attachment. The animals themselves are attaching to the ropes. So if that isn't strong enough, the animals tumble to the ground and they're dead. So it really, so much of the survivorship of these organisms depend on having a strong attachment. And the way they do this, in this one video loop is playing over and over again, they attach with collagen-like fibers, and they mold these one at a time. Each one is called a byssal thread. And to do this, they stick their foot out, and in this particular video, it's pressing against the wall of an aquarium. And you can see there's a depression right here, where it's forming a glue, and that glue is attaching to the wall. And then at the same time, it's excreting other proteins into a groove to make a fiber. So this is a process similar to polymer injection molding. They're molded one at a time, and an animal will make 50 to 100 of these to make an array of tethers to hold them to whatever surface they need to attach to. So just keep in mind that the fibers are not spun like a spider, they're actually molded. Okay, this is sped up, it says 3 times. So to make a single thread takes about 2 minutes. They can make them in a spurt, maybe 5 and 20 minutes. Well, making 20 a day would be a pretty good feat for an animal. Okay, so I mentioned that the attachment is really critical both in natural populations and in farm populations. So I mentioned I had gone to Spain. Here's one of those growers, and what he's showing me is, in a very unhappy face, is that there's bare spots on his lines. So what a grower wants after 6 months or a year is to have a very thick population of animals on those ropes so that they can bring them to market, and they get very sad when they find bare spots. And those bare spots are known as fall off in the aquaculture industry, or sloughing. And those can also happen in natural populations. This one comes from Rhode Island, and that's also a natural process. So I've figured out, at least from these types of populations, 20 to 35% of the population falls off or gets disturbed each year. So it's a substantial form of mortality to the population. So what I was interested in, since mussels are becoming dislodged, and we know that they are subject to mechanical failure, is there a way that we can develop a theory to predict when this happens? Okay, so this is something that Brad was talking about, is once we understand the mechanisms by which these processes occur, then we can start to make inferences about how that process might change when conditions change, if the climate changes or something different. We talk about different species. So I came to this with my biomechanics hat, and it's not that hard a problem. It's really a physics imbalance. And what happens when an animal is dislodged is it has experienced a hydrodynamic force that exceeds its attachment force. So if you know something about hydrodynamic forces on mussels, when waves come and crash on them, and you know something about how strongly they attach, then we could predict this process. So I spent some time doing that when I lived in Rhode Island. I was on the faculty there, and it's not an island, actually. I just thought I'd mention that. It's a really nice place to live. So the way to think about hydrodynamic forces is that waves will come and wash over a set of animals like this. And there's 2 kinds of hydrodynamic forces that they can experience. The ones here in the front are going to feel the brunt of that force. So what they'll be experiencing is something called drag. That's a force that pushes things downstream. But these aggregated mussels, they're actually protected from that immediate hit of the water. But instead what they're experiencing is that the water scoots over the top. And as it scoots over the top, it speeds up. And whenever you have a relatively higher flow, you see a decrease in pressure. And so what happens to these is they're experiencing relatively low pressure here, higher pressure there, and that lifts them off the substrate. So this is a similar force to what keeps airplane wings aloft. Good for airplanes, not good for mussels. So here's just some of the equations for those forces. I'm not going to go into that. The main point is if you know something about the size of the animal and how fast the water is moving, then you can predict these forces. Okay, so to develop this theory then, and I've done this for a couple of different populations around the world, but I'll tell you about my Rhode Island work. First, we need to know something about water velocities, and to do that, I went out and measured wave heights in Narragansett Bay. So that is a bottom-mounted pressure transducer. It's a sensor with a computer, and it's more than happy to think about what the water is doing above it, and it will record 4 times a day how wavy it is out there. So if you know something about wave heights, then you can use wave theory to predict what the water velocity would be when it crashes on the shore. And I knew something about the hydrodynamics from using flow tanks similar to the ones that Brad just showed you. So we can actually calculate the forces that the animals are experiencing. So that's pretty nice. But we also needed to know, as I told you, how strongly attached the animals were. And to do that, it's pretty fun. Actually, you go out at low tide and you can drill a hole through a shell, stick a fishing hook in there, attach that to a force gauge, pull it off, and you've now measured the force in newtons it takes to remove the animal. And we normalize that to the size of the animal to measure attachment strength or the tenacity of that animal. Okay, so those are the pieces we needed to develop this prediction of when things should get dislodged. And I mentioned I did this in Rhode Island, and I did it for many, many years. This is a summary of 3 years of measurements. Both of the waves and the mussel attachment. And what the model, to develop the model, what we learned is that most of the fall off was predicted to happen in 2 specific months, September and October. And this was actually not what I had expected at first, because many of you have lived in New England and know that it's wavy from October all the way through March, right? There's lots of storms that come all during the winter. So, and indeed, during those 3 years, we did see this as well. But it turns out that those storms came at a time when the mussels had very strong attachment. They were able to withstand that challenge. The time that they're really in trouble is right here at the end of summer. They have very weak attachment, so even the slightest storm can come in and cause this big mortality event. So this was a prediction, this was a model, and I wanted to actually test it because, you know, otherwise you just have models that make cute stories, but it's always nice to prove that they work that way. And so to do that, I went out to the field again, and I tracked the persistence of populations for those same years that I was developing this model. So this is one of my study sites, Black Point and Narragansett Bay, and I know it looks really beautiful there. And we were sampling in the summertime when it looked like that, but we also had to do this year-round because I needed to know what was happening in the winter. So this picture is of that same site after a snowstorm, and it illustrates nicely actually where we were working. So, and I have to say it wasn't me, it was a technician, went out and found the 2 bolts where we had our transects, and he had to pour seawater over to get rid of the snow so he could take pictures of this population. So we took pictures like this for 3 years. This is what they might look like, a small, the size of your notebook, but we had many of these pictures, 40 of them. We went every 2 weeks for 3 years. And then we asked in each one of those, what was the percent cover of animals? And so this is about 100% cover. This is more like 40% 2 weeks later. So this is the big blowout, right? That's the big disturbance event that we were chasing. And so the question was, when do we see those disturbance events? Is it really going to be in September, October, like we predicted? And the answer is, so black is my predictions and the gray is my observations. So indeed, it's not a perfect fit, but it's pretty good. 80% of the variation in mussel dislodgement from month to month is predicted by this really simple biomechanical model. And importantly, I predicted that there would be no dislodgement here in those months where historically we typically have big storms, and indeed I didn't see any dislodgement. So the animals were strong enough to withstand that challenge. Okay, so the punchline here is there's specific months, the timing matters, that there's only specific months that the mussels are really at risk. And so developing that mechanistic model was the only way I could have come up with that. And I wanted to explain a little bit more about why this is important. So those of you who live in New England, you're aware that there's many big storms, and you might have thought that any storm, anytime, would cause the same amount of damage. But it turns out the ones that do the most damage are these early season storms. They tend to be hurricanes. So Hurricane Sandy in 2012 came in. That's a tropical storm making its way up the coast. So that has big waves. And it's a problem because it comes in October when the animals are not very strongly attached. There was a lot of dislodgement that year. So that very same year, there was another big storm. This was Winter Storm Nemo. That's an extratropical storm, so it's a different type of storm system, but it generates big waves nonetheless. But that's the one that doesn't cause much damage to the mussel population. The reason I'm making this point is there are very different types of storm systems. We know climates are changing. We know that the thermal circulation of the Earth is changing. We know that certain storm patterns are going to be changing over time. And so one of the predictions with increased warming in the climate is an increase in the frequency and severity of hurricanes in particular. And this is a real cause for concern, not just here in Florida and for the whole seaboard, but for mussels in particular. That's the type of storm system that they're going to be really vulnerable to. So if instead the prediction was a weaker hurricane activity, or increased of these other type of nor'easter storms, then that we would predict that would have less of a impact on mussels in New England. Okay, so that's just an illustration of why understanding the mechanism matters. Okay, so why does this model work so well? I'd like to think I'm pretty good at measuring water velocities and wave heights out on the shore. And actually, when I started this work, I thought that would be the most important thing to keep track of. But it turns out it's all about the animals. What is driving this pattern is this strong seasonality in attachment strength of the animals. So they are twice as strong in the late winter and early spring. They just crash every summer. So what I've spent most of my time the last 10 years is trying to understand what in the world causes this. And just to keep it short, there's many ways an animal could be weaker. They could make fewer threads, they could make a different quality of thread, or those threads could be rotting faster. Something about the environment could be changing it. And all of these do matter, but what I'm going to focus on is this thread quality issue. Okay, so this is when I moved to Washington State, and I was armed with this knowledge that mussels get weak. And I really didn't know what was causing it. I knew all kinds of weird things were happening in water in the summertime, and I've been working through to figure out which ones are the most likely culprits. But here's my list of the things that I needed to worry about. One is water temperatures warmer in the summer. There's less food in the summer. There's differences in the oceanography. The animals are spawning. They don't have as big a body weight. So all of these energetic and environmental factors might influence how these animals are behaving. So on top of that, that's when ocean acidification came on the map. So that's yet another environmental concern that we all were thinking about. So this is just the Keeling curve. I think Brad already showed this data, but Charles Keeling is the one who measured atmospheric CO2 here at Station Mauna Loa. And then these data are from the HOTS transect, the Hawaiian Oceanic Time Series. And so what they were able to do is we know that atmospheric CO2 was increasing, but these data are really important because they show that seawater partial pressure was also increasing at the very same rate. So that means the atmospheric CO2 is getting into the seawater. And whenever you see increased PCO2, you see a decrease in pH. So this is really the evidence for ocean acidification, that decline in pH. So we were also worried about this with the mussels, and we got funding from the National Science Foundation to develop this laboratory at Friday Harbor Labs, specifically to look at the effects of ocean acidification. And so we set up these coolers. They really are igloo coolers, but they are isolated units in which we can control the pH that the animals are experiencing, the water temperature. We can vary the food supply, and we can put any organism of interest in there and wait a little bit, and then come measure whatever parameter that you're interested in. So some people use this facility to measure metabolic rates. Some people measure it for growth rates. I, in particular, was using it to measure the effects of ocean acidification and temperature on Mussel threads. Okay, so what we would do is put animals in these buckets for anywhere from a week to 4 weeks. Oops. And harvest their Mussel threads and then bring it to my other laboratory here. And this is the torture machine. It's called a tensometer. It's a materials testing machine. What you're seeing is a tiny fiber in some grips underwater, and we will pull them apart slowly to measure the force that it takes to break them. So we're measuring how strong they are. And to give away the punchline, what we've learned from these experiments is that there's lots of ways that mussels can lose their grip. So we know that environmental conditions weaken mussel Mussel threads. Specifically, we've been able to show that a low pH is going to make the attachment weaker. So they're fine in relatively ambient PCO2 conditions, like this is the current day conditions, but as you ramp up the amount of carbon in the water, which means you're lowering the pH, then we see about a 40% drop in attachment strength. If you convert this isn't PCO2, but this really means a critical threshold of about a pH of 7.6. Okay, we also know that high temperature is a problem. In fact, it's even more of a problem than pH. We see a much more dramatic drop in how much force it takes to break a Mussel thread. It's about a 60% decline. And in this case, the magic temperature for this particular species seems to be 18 degrees. Okay, so I just want to, just remember those numbers, but I want to take a moment to say why we think this is happening. In the case of the pH effect, we actually know quite a bit about mussel adhesives, and part of this is due to the interest in preventing them from attaching to ships. So it's research not done by myself, but other biochemists, and a lot of this was funded by the Navy. And so we know that there's very specific proteins that are secreted in that depression of the foot. Remember I said it was pressed against the wall of the aquarium? Well, you can make them attach to pieces of mica and probe what's happening with the proteins that they're secreting. So this is work done by Herb Waite and colleagues, and what he's been able to show is in that distal depression, when they're pushed against the hard substrate, he's been able to show that the pH in that vicinity is about 2 or 3, really low pH. So somehow that animal's able to pump protons and lower the pH. It's actually really important that it does this because that's the only way to keep all those proteins in a liquid form to get to their destination. So if the pH were higher, they would bind and never actually set up where they needed them. Okay, so to deliver these proteins in a liquid form to the place they need to go, they need to lower the pH. And they do that pretty dramatically. So what that allows is for this L-DOPA molecule to actually attach to the surface. Okay, so then the animal takes its foot away, and seawater pH is usually around 7.8 or 8. So what happens is when the foot goes away, we're going from a really low pH to a much higher one. And that allows other linkages to form between in these molecules. In particular, in the presence of oxygen, these molecules now can attach in this direction and make a structure that can resist force. So what happens is when the animal first releases the glue, and it's not set up well, not very well cured, just like an epoxy would need to cure over time, you see a milky white fiber, and it takes several days, up to a week, for that fiber to fully tan And yes, it becomes tanner, but what's happening is crosslinks are forming and giving structural integrity to that adhesive. Okay, and what I've just told you is it's pH sensitive. So what my graduate students have done then is to have animals make their glue and then adjust what water temperature they're curing in. And so if we give them the pH of 8, which is what normal seawater would do, over time they get stronger and stronger and stronger. So initially when they're white, they start out down here, and as they tan, they get stronger. If we manipulate that seawater to lower the pH, and this could be a pH of 7 or 5, the adhesives are weaker, and what we believe is happening is those crosslinks aren't forming as effectively. Okay. So that's not a big difference here. pH of 8 to pH of 7 is in the range of what we're seeing naturally in our seawater. And we think that just these types of changes can dramatically weaken the animals. Okay, so that's all laboratory experiments, and those are pretty fun to do lab experiments, but I always like to go out in the field and ask, well, do we ever see these conditions anyway? And this has been a really exciting part of my work, something I've always wanted to do, and I've gone to where there's a lot of mussels, and it turns out that that's a mussel aquaculture farm. They have lots of mussels for us to work with. So we have partnered with Penn Cove Shellfish. They're the oldest and largest mussel growers in the United States. Ian Jeffords is the president of that company, and he's been really supportive of our work and really facilitated my graduate students getting out onto the water, setting up equipment. So I like to say this is truly a partnership. We're the academia part, Ian's the industry, and we've also gotten state and federal agencies involved. And I want to emphasize I could not do this alone. One is that we didn't have the manpower. Two, Ian and his engineers have helped us develop welding different frames for us to put out on the water. They really know how to get things to work on a floating raft much better than we do. And the other agencies have helped us get by the instruments to take these measurements. Okay, so what I'm showing you here is my graduate student, Laura Newcomb. She's holding a data sonde. It's a YSI data sonde, and it measures many aspects of the water temperature, pH. We're also measuring food supply and oxygen. So what that data sonde does, unlike the one that I had down in Rhode Island that just was stuck at the bottom of the ocean, this one is cabled, and that data in real time is being telemetried to the mainland, into the main office of the Penn Cove Shellfish, and then that's connecting to this NANUS website. So when I say federal government, this is a group that's supported by NOAA. And what it is a platform to observe data real time. So this is pretty special that anybody can, this is open to the public, right? So anybody can look. And so rather than wondering whether our meters are working anytime, in fact, I was just looking at this yesterday on my own phone, so I have the app, and I can ask, gee, what's the pH right now in Penn Cove? And I can tell you, it's pretty interesting. Okay, so this is what we do. One is that we're getting real-time data. Two, we know whether our instruments are out of calibration or whether they're just plain not recording. That's really valuable. The other reason this is a really valuable enterprise is that we go out on the water, and Ian told me the other day, because I said, I love this website, I love this app, it's really great that I can see the data. He goes, well, actually, so do my workers. So what you're seeing is during a lunch break, sometimes they're just checking their email or texting somebody, but what Ian says is it's not uncommon for them to get on the app. It's because they're on the water every day, and they notice some days the water is a little bit greener, or maybe the animals look a little bit different. And this has just the fact that we're out there and talking to them and talking about our data, now they're actually curious about it. And I really like to think that this is the win-win-win part of our study. We're not there disseminating information at them. They're the ones that are really helping us with our research. I can't emphasize enough that the growers are the ones on the water every day, and they're the best observers that we have. We only show up there once a month to collect some data. So many times they have given us ideas on how we should maybe modify our research. I bounce ideas off of them all the time. Okay, so I think this partnership aspect is really important. Okay, so do we see low pH conditions in the field? The answer is yes. So here's one of these traces. The line in blue says that's the danger zone. Anywhere below that, we're going to call that low pH, and the glue might not set up very well. So, but that's at the surface waters. We had another data sign down at 7 meters. And it was like night and day what's going on. And this was quite the shock. That just in the 6-meter range, really the difference between, you know, somebody who's a really tall man, right? At the head, you're going to have, for the most part, pH waters that are just fine. But down at your toes, it's, for the most part, quite acidic. So what we have done then is continued these measurements, and I'm not going to go through this in too much detail, only to show you that we've measured pH, oxygen, food supply, this is chlorophyll, salinity and temperature, and attachment strength of animals. And we've put this together to see if we could predict what conditions are most important for predicting weak attachment. So to summarize that, here's the answer. The two factors that came up in our analyses as being the most important are pH and temperature. In fact, temperature was more important than pH, but they both matter. And this is a 3D or contour map summarizing our data. So first focus on this square here, and it's color-coded to tell you what attachment strength would be under the certain condition of temperature and pH. So for a temperature of 8 degrees and 8.2 pH, for example, then you'd be in a very strong condition for the animals. So anything that slides you down the surface into the dark blue region means the animals are really weak. So that would be bad for the mussels, bad for the growers. Okay, so then I've put on these triangles are the data we've collected at the surface in pink and at 7 meters in yellow. So you can see there's not that many times when we have a strong attachment. And for the most part, it's the ones at the surface that are doing really well. But they're also half the time for these pink points, they're in this bad condition. And what's pushing them over to this dark blue zone here is high temperature. So animals at the surface aren't prone to low pH so much. What makes them weak is the high temperature. In contrast, the animals that were held at 7 meters, first of all, they're weaker overall, but there are times when they're strong, and when that happens, it's because the pH has changed. It has very little to do with the change in temperature. So just to summarize what this means is both low temperature and high pH matter, and they both have to be correct for an animal to have a strong attachment. So it's either high temperature or low pH is going to send them into this danger zone. Okay, so what I just told you about is mostly Middleus trossulus, the native mussel. There's other mussels that are grown in Penn Cove and in Washington State, and they occur there naturally. What I've told you already is that ocean warming weakens Middleus trossulus, and if we look at attachment strength as a function of temperature, this is this blue line, that this particular species gets weaker when the water conditions are above about 18 degrees. The other species, now I haven't talked much about them, but they're the Mediterranean mussels, warm Mediterranean water, right? And they have become established on the west coast of North America. They're more common in Southern California. They're kind of making their way north. And it turns out this particular species loves warm water. So they're indistinguishable in their attachment strength at these more modest temperatures. But when you get above 18 degrees, that's the one where trossulus crashes. Middleus galloprovincialis starts making more threads, better threads, and more durable threads. So they have a much stronger attachment. So we can take this information and think about if we were going to say that attachment strength is what determines survival of a population. What will happen? Can we use this information to predict current distribution of the animals? And so what Laura did is to go, this is my graduate student Laura, go look at sea surface temperatures and ask where is that 18-degree threshold happening. So here in blue, these are waters where they don't often see 18 degrees exceeded. And so we would predict that for all kinds of reasons, Middleist trossulus would be favored. That's the native species. But if seawater conditions get over 18 degrees, and that's what's happening down here in red, then we would predict that trossulus would be weaker and that there would be an advantage to this invader. And so this is simply color-coded based on what water temperature you see there. And so it turns out that this, where we would predict that Gallo provincialis would be favored, corresponds pretty darn well with their current distribution. So what Laura did is ask, well, what's going to happen with climate change? Our oceans are warming. What's predicted for the next 100 years is a 2-degree warming. And so now where do we see that 18-degree threshold happening on our west coast? And the answer is that we would expect the non-native species, Gallo provincialis, to have a huge range expansion all up and down this west coast. And it's not just on the outer coast. There's a bit of a change here around in the Salish Sea or the Puget Sound as well. Okay, so what I would suggest then is, what are we going to do about this? Let me back up a little bit. So when I think about Ian Jeffords at Penn Cove Shellfish and what does the future look like for him, he's growing both species right now, trossulus and Gallo provincialis, but I think what the future is holding is shifting to a different species, more emphasis on the warm water species. Okay, so just to summarize, what we've been doing lately is trying to link our lab studies with field observations. And these are some of the questions that we're still chasing and why. But we're still trying to identify specifically what triggers fall off. We're pretty sure temperature and pH are involved, but we can't rule out food for reasons that Brad has explained also. So we need a longer-term data set to really tease that apart. One of our goals is to develop adaptation strategies for our growers so we can use, with our real-time data, this is now casting, so the growers can look at the water conditions live and know when they're in dangerous conditions, and they can actually modify their practices. So they can choose when they take all the animals off the lines to readjust the densities. Maybe don't do that when water conditions are bad. And they can adjust some of their harvesting practices as well. And then not just with nowcasting, but it's really useful to develop these studies for forecasting for future hypothetical conditions. I just gave you some examples with just water warming. And all of this will help us help the growers make informed decisions about which species they would grow in which locations. So this work has been really valuable. I've talked a lot about it in Washington State and in other parts of the country. And it did get the attention of our governor. This is Governor Jay Inslee. And he came to visit us at Penn Cove, and he wanted to hear about our research. And a couple things that I learned is that Jay Inslee is really smart. It didn't surprise me that he would know a little bit about the research, but I was really impressed with the level of knowledge that he had. I know he's a busy man and can't read everything, but really asked some really key, pointed questions. The other thing I learned is he travels with tweeters. These two women were there, and they're looking at their phone. They're taking pictures and typing and getting the information out. So, when Jay Inslee tweets, it's not really him. So this is the question. I'm telling him all about our data sons and all the different things that we're measuring, oxygen and chlorophyll and such. And here's the question he gave me, and it's a whopper. But it's actually the right question. He needs to know. He's a politician. He needs to make policies that matter for the next 100 years. And so he's asking me, should we worry? What will our oceans look like in 100 years? And I have to admit, and I think most of the scientists in the room, we're really uncomfortable answering that question. We like to waffle, and wither around and say, well, let me just collect some more data and then I'll tell you. But we can't do that. That's not good enough. We have to be able to answer these questions. We've now gotten to the point where the politicians, the business community is interested in hearing about these problems and coming up with solutions. So yeah, I tell them, yes, we should worry. We're already seeing some of these corrosive waters currently at Penn Cove. And we know that when they hit these extremes, the animals are going to tumble off the ropes. The concern is that we have a shifting baseline, and things are ooching warmer and ooching to more acidic conditions. So when the frequency and severity with which they hit these bad conditions are just going to increase. So yes, I'm worried, and I think the more we can do to get the word out to the business community and the politicians, I think we'll be better off. Okay, maybe George will show that picture. So in closing, I just want to thank many graduate students and other collaborators and my funding sources, and we'll take questions in a bit. Thank you.